We are recording now. So it's my pleasure to introduce Juan Gallego. Juan is a senior lecturer at Imperial College London. That's the equivalent of associate professor in the metric system. And
Imperial system.
No, metric system. Yes.
It doesn't quite map.
Yeah, it doesn't quite map. He's interested in neural control of behavior at the level of basic science, but is also interested in the engineering applications like BCI. So with that, I think we're going to start the discussion today. Yeah.
Okay, thank you for the introduction and for having us. I'm Juan.
Yeah, I'm Joanna, one of the co-first authors. I'm here to answer questions, but I'll let Juan lead the way.
Yes, I have to. Okay, so I'll share my screen and we can get started, I guess.
Yeah.
Okay, so yeah, so we'll be, like I guess Joanna will be talking about this paper that Joanna co-led with Mustafa Safai, a postdoc in the lab, and that I kind of co-supervised, if that is the best word, with Matt Perick, who may pop up like randomly later. Also with Juncho and Joss from Janilia and Lee Miller. You know, I go through a paper, but I thought like I would start with a bit like zooming out and the ideas that led to this paper ultimately. So I have a few slides on how we approach this problem on understanding how the brain controls movement. And basically what the level of description that we're interested in is something, you know, that is kind of in between the classic interest in single neurons. And here, of course, I have a drawing of a caja neuron as a good Spaniard. We always have to put a caja neuron in our slides, and behavior. And the level that we study is the, you know, interacting neural populations across multiple brain regions. The way we do this is by, instead of focusing on how the activity of each neuron relates to behavior, we make explicit that, you know, due to a number of biophysical constraints such as neural connectivity, neuromodulators, common synaptic inputs, neural interactions, etc., the activity of neurons, like neurons tend to go very according to specific patterns that I'm trying to represent here in green and blue. So and that we call neural modes. So a bit our whole philosophy is that the brain works not by modulating the activity of single neurons independently, but by modulating these population-wide activity patterns that we call neural modes. And, you know, this sounds very vague and philosophical. So the way we can use this idea experimentally is like if you imagine that you've recorded from, for example, these three neurons, instead of looking at the independent activity as a function of time or task parameters, what we do, and many other groups do, of course, is like build this neural state space in which each axis is the firing rate of a neuron, essentially. So then in this space, the activity of these three neurons, for example, when an animal is about to move, becomes a point. And as the animal moves, for example, the arm, this state will describe some trajectory. And now what you can do is study the trajectories that the activity of these neurons define during a task, like I will do later. But an interesting observation that holds from the locus olfactory system to human prefrontal cortex is that these trajectories remain confined to a lower dimensional part of this state space, which is what we call a neural manifold. And that, luckily for us, is defined by the dominant activity patterns of the population that are these neural modes. So a lot of the work we do in the lab, and again, that more and more people in the community are doing, is trying to understand the neural basis of behavior by focusing on the geometry of these manifolds and the activity within them that we call the neural dynamics. And I'm going to be only focusing on this study that you guys invited us to present, but here are a few references with reviews with many studies using these ideas from us and other groups. As a quick I guess like PR moment for people who don't know our work. And since this is going on YouTube, I wanted to flag a few things that we've done in the lab or we are doing with this idea, like gaining some insight into commonalities in neural activity across behaviors that you cannot see because single neuron tuning changes a lot. We're also able to use this idea to uncover that or to show that the brain uses the same latent dynamics when a monkey is doing the same task in different years. But the interesting thing here was that you could uncover this with different neurons. We also have some work on the neural basis of learning to motor learning to counter different perturbations. This was led by Matt. Now in the lab, we're starting looking at comparing the geometry of these manifolds across regions. This is Katya's work on bioarchive. We're also looking at the intersection between movement planning and more decision-making or cognitive aspects, such as making sense of sensory cues. And we are also interested in the relationship with other population activity patterns like LFPs, as this is Cecilia's work. This is, for the discussion or for the future synthesis in YouTube. Now we will start with talking about this study. And the question we wanted to ask is a very, I guess, obvious one, is, and I want to introduce it with an experiment, as I always do, because we are also experimentalists. So I want you all, and people at home too, to close your eyes and touch the tip of your finger, of your nose, with the right index. And leave it there with your eyes closed. And now open your eyes. And you could all do this, right? So this is something that is obviously trivial. By the way, I didn't see anyone doing this. So, it's obviously trivial that we can all do the same thing, but the question is, how come that we can do the same things if, our brains at the cellular level are different, like at the single neuron level are different, right? Because each of our brains is unique. So if we start from the beginning, and the reference for this is wrong, by the way, it needs updating. The behavioral repertoire of a species has developed over evolutionary timescales, right? And what this ultimately implies is that the information for the behavioral repertoire of a species is encoded by the genome that has instructions to build brains of a certain class. So, for example, these are all the monkey brains, as you can see in the image. And the thing is, even though each of these brains is unique at the levels of single neurons, the way that these neurons are organized in microcircuits, layers, regions is the same, right? Because this is what makes a monkey brain a monkey brain. So now, and this is why I have to give the introduction before. If you remember, I said that we think that these neural manifolds are shaped by circuit connectivity patterns and other biophysical properties. So if you believe that all these biophysical constraints are common across the individuals from the same species because they are specified by the genome, then it follows that the manifolds that these brains can develop should be the same across animals from the same species. Okay, does this make sense? Okay, so this is of course very vague, but it leads to a testable prediction. That is that if you have two monkeys, and if it's indeed true that each of their brains is an instantiation of this neural landscape, if you record neural activity as they do the same task, then you should see the same population dynamics, even if the neurons that you are looking at are obviously different. So what I mean by being the same is that you can use simply like a method that linearly transforms these sets of population dynamics. So basically it rotates them and stretches them to show that they are the same sets of dynamics. So the intuitive way to think about this is that the blue monkey is my left hand and the green monkey is my right hand. And that, if you look at them like this, you don't know if they are the same object. But if you rotate the two, you can actually see that they are. Okay. And we did this using a method that is called canonical correlation analysis or procrastis, but these are simple linear methods. I should also say that our hypothesis leads to a second prediction, that is that as the behavior becomes more different, these dynamics will become more different. Okay.
There is a question, I think.
Yeah. Well, I always ask this question. Procrastis has only rotation but no scaling, or differential scaling for the different axes. CCA is rotation, but allows differential scaling along the different axes. Now, in the Nature paper, I guess the way I read it, you use CCA. How different is the scaling along dimension 1, 2, 3? I would, from my perspective, when we do very similar things with fMRI pattern, and then we usually do not allow for different scaling along the different dimensions because I think, in my understanding, the neurodynamics really changes whether that second dimension, the rotation is really a circle or it's a very stretched oval, right? I mean, the noise robustness and so on, to me, is very, very different across these two systems. So I wouldn't call them the same manifold, or this, well, maybe the same manifold, but not the same dynamics, right? I don't know. How you think about this?
I guess I'm not too sure what you're saying about the specific scaling. I don't know if we've measured this specifically, but if we have a lot of examples in the supplementary figures as well that show once you rotate them, they look very similar. And in the supplementary
Once you rotate and scale, or rotate?
Rotate and scale. But we also
you scale each dimension separately, right? You allow a different scaling factor for each monkey for each dimension, meaning like you could make a rectangle into a square.
And with CCA you could, but not with Procrastis, right?
Right.
Which is why you asked.
That's what I'm asking. Excellent.
Yeah.
And that's why in the supplementary figures we also ran all of our the major of our analyses on Procrustes as well, to show that it's also robust with Procrustes. So yes, we use CCA, but I don't think it's specific to the method that we used.
So that would indicate that the differential scaling along different dimensions isn't a big factor in your life.
Yeah,
Right.
that is, if you look at, I can try to find the figure later, John. I think it's on the supplementary slides, especially with you being here. I was expecting this question, but I think if you go to the supplement, it's like we did Procrastis and then we calculated the correlations the same way we do with CCA, and maybe they were at 85% as high, something like that. So this suggests that, right, like Joe, like they were like 85%. So this suggests across the, I think is the mean across the first 4 dimensions or something. So this suggests that there's not that much stretching.
Okay.
Yeah, of course you can take the opposite approach and use like fancy neural networks that will do everything much more similar, right? But that was not our goal here. Okay, any other questions?
Not for now, I guess.
And what is what is the dimensionality reduction technique? Is it PCA? Is it factor analysis?
Yes, we use PCA and then we perform CCA on the PCA.
And to add on that, like back in the days, we did some comparisons between PCA and factor analysis. I don't know if they are published on all the papers. I know that at least on Matt's neuron paper on learning, we did the entire paper with factor analysis and PCA, and all the results were qualitatively similar, and almost quantitatively as well. So in our hands, we don't get a big difference between the two.
Mm-hmm.
Okay. So I'll move on if there are no more questions.
Yeah.
Okay. So so the task that I would be focusing on for a great part of the task is the task that is kind of arguably like helping solve motor control, because we are all obsessed with this center out reaching task. So this is monkeys that have been trained to move this joystick, or humans. Basically, they move the yellow cursor to the target to get a reward, and then they come back and targets are presented, 8 locations, and they are presented randomly. So what I'm showing you here is the position of the hand from 8 trials from 2 different monkeys, and the arrow indicates the direction of the target, and the 2 colors are X and Y axis. So as you can expect, since these monkeys were like pro center out athletes, because they have been practicing for years, their trajectories are very good, very similar, and very consistent also. If I show you neural recordings, of course, these look different because, there are different brains, different numbers of neurons. But to us, this doesn't matter, right? So here, if you now do PCA and look at the latent dynamics in the first 3 dimensions of this PCA manifold, and I'm being a bit loose here with the use of the word manifold, we can talk about that too if you want. So these are the trajectories of these populations when the monkey reached to the monkey C, reached to these 8 targets. And these are the trajectories when monkey M reached to the 8 targets. So now the moment of truth for the paper is whether we can basically align. So monkey C is my left hand and monkey M is my right hand. And can we uncover that they are the same? And you can imagine that is what we found. Otherwise, we wouldn't be able, we wouldn't be talking about this paper, right? So this just shows that these 2 sets of squiggly lines look very similar. And I should say that we are aligning single trials So, it's not that we are optimally aligning the trajectory for each trial. We concatenate all single trials from one session and we align them. And we can quantify that, for example, by plotting the correlation per axis of this manifold, per neural mode. And you can see that the first few are very high, then they go down, and this is how CCA works. But of course, these numbers, 0.9 looks pretty good for neuroscience, but it doesn't mean much if you don't compare it to upper and lower bounds, right? So we did a lot of lower bound comparisons on the paper. This is just taking random epochs and trying to align across monkeys. So not very challenging, but there's many more in the paper. But the interesting comparison is the upper bound. So the upper bound that we took is, now we're going to take data from only one monkey during the same session, looking at the same neurons, and I'm going to align this subset of trials within monkey, within session, within neurons. And what you get is this gray line. So you can see that the latent dynamics from two different monkeys are almost as similar as this within monkey upper bound. For this example comparison, and this was the case for many data sets from 3 or 4 different monkeys, 3 different monkeys. So, this suggests that these latent dynamics are preserved across monkeys doing the same task.
Now, the within, is that different cells, or what recordings did you use here? Like, is it
same cells?
Same cells, so it's like a array implant or something.
Yeah, it's Utah arrays. So this is all the data and all the monkey data is Utah arrays. Okay. And many of these data sets, we were being very careful. So are spike sorted neurons that we could track during the entire session. So it's same monkey, same day, same neurons. And of course, the next thing we did was like whether these aligned dynamics were relevant to behavior. And by this, I mean whether we could decode the kinematics of the movement, right? That was our readout of behavior. So here you can see the hand position for 8 trials performed by one of our monkeys. We could predict very well within monkey, as people have been showing for decades. But the cool thing is that we could train a decoder in one monkey, and it would work equally well on a different monkey. So you can see that the blue predictions are all extremely similar to the gray predictions, so across monkey to within monkey. And of course, if we didn't do the alignment, many of these predictions were terrible. So these are the example trials. This is the summary for this particular comparison, and this is across our 120-something comparisons. So in general, we could predict across monkeys virtually as well as we could within monkeys. And this is continuous hand kinematics. Okay? So this suggests that these preserved latent dynamics are informative about behavior. Now, we wanted to extend this result to another data set from our collaborators, Josh and Juncho Park from Janilia in the mouse for 2 reasons. One is to show that this result generalizes to mouse, which is interesting. But the second one is that these were mice, performing a very cool task, and I should have added the reference to the paper here. It's on bioRxiv, the first author is Juncho Park, where we explore this task a lot. So these are mice that have learned to reach, grasp, and pull on a joystick that can be either here or here, so 2 positions, and also 2 different locks. And Juncho trained these monkeys for these mice for a few weeks. So you can see, if you look at the velocity of the hand at the bottom, is less consistent across mice that the hand kinematics were across monkeys. So we have more behavioral variability across monkeys than across mice and across monkeys. Sorry. So this is the correlation in kinematics within mice versus within monkey. So why I'm showing you this is because this allows us to begin testing the hypothesis that the correlation, the preservation of latent dynamics is linked to the preservation of behavior. So now our prediction would be that since the behavior of the mice were less consistent, we should see latent dynamics that are less preserved. Okay? And that is exactly what we found. You can see that in this case, the across mouse correlations are still like well above the control, but they are in between the lower bound and upper bound controls, right? Which was not the case for the monkeys. And we can look at this more like directly by basically plotting the correlation in latent dynamics versus the correlation in behavior for each session. And if you look at this plot, you do see like a nice association between the two. Like in the monkeys, there's not a lot of variability, but in the mouse, you can see that the more similar the behavior, the more similar the latent dynamics. Okay, and the same is true for the decoding. The more similar the behavior, the more similar you can decode across animals. Okay. Okay, so I know that some of the skeptics in this room, and I know at least a few people there that are skeptics for good reasons, or which is a good quality. Maybe wonder like these tasks are very stereotyped, right? Because in the monkey task, we are basically looking at reaching in 8 directions. The mouse is only reaching forward to the different locations and then grasping against 2 different forces. So what we did to push this idea further was look at a task where monkeys had to produce a sequence of up to 4 reaches to targets that were popping up as they were hitting one. So they would see one target, they would hit this target, and then another one will appear immediately. So they were making a lot of errors, and there's a lot of variability. And this is, you can think about it as the monkey version of whack-a-mole. So it's basically monkeys doing this with the joystick. So these are the kinematics of 2 different sessions from 2 monkeys. So you can see that there's reaches all over the workspace. This is representing the entire workspace. But actually, Joanna went through the pretty labor-intensive process of developing a method to match reaches across monkeys and to obtain things like this. So what you see is like the solid and dashed lines were the reaches that are more similar in terms of hack kinematics across sessions from these monkeys. And the cool thing is that, again, we could align the latent dynamics across monkeys very well. We're a bit farther away from the upper bound than we were for center out, as we expected. But still, like, we can show preservation of latent dynamics even when there's not a structured task, if you want. And this is one example, but again, this was the case across, I think, 11 comparisons or something like that. No? From 3 different monkeys. And by doing this, we could also do another check that we're curious about, that is what happens to our alignment. If we have, like, let's say one day we have 100 conditions instead of 30 like we had, will our ability to align go down and down, or will it, or is it independent of the number of conditions? So if you look at this, it is true that with very few reaches, we align a bit better, but after you have like 5 to 10 reaches, the ability to align remains the same. So this suggests, like, if you believe in extrapolation in neuroscience, if you have like 100 conditions, we should still be able to align, even if this task is much less stereotyped. Yeah, and one more thing about behavioral similarity and the relationship to the behavioral preservation of latent dynamics. So far, I hope that I and Joanna, with her answers, have convinced you that when 2 animals are doing the same task, they generate the same latent dynamics in motor cortex, and that these seem to be related to the similarity of the behavior. So we wanted to put like a final nail on this coffin to the extent that we could by comparing the similarity of latent dynamics between 2 monkeys doing the same task to the similarity of latent dynamics from the same monkey doing 2 tasks that are very similar. Okay, so we look at data from one of the papers I flagged at the beginning, where monkeys were doing a variety of risk tasks like this. So you can see that these were 3 tasks with the same targets on the screen, only 1 degree of freedom. So the forearm was restrained like this, and the monkey was in the isometric task, was basically only generating force without moving. In the movement task, it was moving the joystick like this, if you can see my hand. And in the elastic task, it was moving the joystick, but there was a spring pushing the hand back to the center. So the EMG looks a bit different. And actually, the velocity of the hand is a bit slower because, there's a spring pushing you back. So now we can see what happens to the latent dynamics if I compare, again, within monkey, within session, looking at the same sorted neurons from these 2 different tasks that are still very similar, right? So here you see the extent of preservation of latent dynamics across 2 different monkeys in the reaching task. And if you compare the latent dynamics within monkey between this movement and elastic task, they are slightly more different than they were across monkeys doing the same task. The same was true for all the risk tasks, and even for another series of reaching and grasping tasks that we looked at in this paper. So to me, this is the most convincing analysis, and I remember we're very happy with these results because it shows that even if you are looking at different neurons and different brains, you see more preserved latent dynamics than if you look at the same animal doing two things that seem very similar. Okay. Any questions?
I miss it on the last graph when you say, yeah, so the black line, the highest line is aligned across monkeys within tasks. That makes sense. Aligned across monkeys. Aligned within monkeys, what is reaching task across any pair of tasks, basically?
Yeah,
No.
so yeah, any pair of tasks. So I separated them in three categories. One is this movement and elastic are the most similar ones in terms of kinematics. Then the risk tasks would be all the risk tasks, for example, comparing isometric versus movement, and like all these debates of force control versus velocity control.
Yeah.
And then the reaching.
The last one, aligned within monkey reaching task. What is this? The one.
Okay, yeah. Yeah, I didn't describe those things here. So these were two tasks that actually were performed by Chris Etier, a postdoc in Lee's lab, who is now a PI in Laval. So not very far from you for Canadian standards, I guess.
Yeah.
So these tasks are, one was like basically in both cases, the monkey started with his hand in this like hand rest that is kind of drawn in gray. And then they had a cue, and in one set of tasks, the one that is shown there, in one task, they had to reach and squeeze this tube that was a force sensing device. So he was reaching, grasping, and squeezing this tube. So it was like a reach and grasp task. And the other task was reaching, grasping a ball, and leaving this and dropping this ball somewhere else. Which makes sense because these are less similar, so the correlation in latent dynamics are less similar than in the risk tasks. Okay. Okay, any other questions? Okay, so I guess to start wrapping up, we were also curious to see, because so far we've only looked at motor cortex, which is in primates and arguably in mice is, well, at least in primates is the main motor output, controlling the spinal cord. In mouse is also necessary for this reaching and grasping task, because if you inactivate M1, they cannot do it. So we're also curious if our results will extend to other areas that are important for behavior, but are not as tightly linked to the output as motor cortex. And we could do that, again, looking at Junchon and Jose's data, because they recorded with neuropixels from both M1 and dorsolateral striatum, which is the mouse homologue of putamen. So the main input of the basal ganglia that receives inputs from motor cortex. So we did the same analysis, and we could align these latent dynamics across mice, and the correlations are very similar to those we found in motor cortex. And we could also decode across mice. So if you remember, our ability to decode across mice for motor cortex was a bit lower than for monkeys, as you can see here overall for striatum. But if I color code these individual comparisons by the correlation in behavior, you can see that as for motor cortex, when the kinematics of two mice become more similar, then our ability to predict across mice also becomes more similar. So it's basically the same results for mouse basal ganglia or dorsolateral striatum as we found for mouse motor cortex. Okay. And the last thing we looked at is that, animals not only move, but they also engage in what we define as covert behavior. So, for example, I have this cool picture of my dog judging me while I was giving a talk during Zoom lockdown. So I felt it was appropriate for now. And, she was probably thinking that I was very boring. But the interesting thing is that we could test whether two monkeys thinking about the same thing, or more precisely planning the same movement, also generated equally preserved latent dynamics. And we could do this because in the center of reaching task, there was an instructed delay period in which monkeys saw the cue, but they couldn't move the joystick, because if they did, they would get a penalty and would have to wait a lot of time for the juice. So they became very good at waiting for the targets. Okay. And while we did the same alignment analysis, you can see that again, we can align these latent dynamics across monkeys very well. And we could also train a Bayesian classifier to predict what what movement the monkey was going to execute later on in the future. And these classifiers worked across monkeys as well as it did within monkeys. Okay, so and I think this is an important result for a number of reasons. One is that it shows that our findings could extend to cognitive tasks, and in fact we are like extending this to some decision making tasks. But the other thing is that it also shows that it's not only due to the fact that motor cortex and striatum are getting the same sensory feedback because the movement is the same, right? Because in this case, there's no movement, there's no feedback input. Because we're only looking at the planning phase. And now my last slide is like to go full circle. At the beginning, we framed this, or I framed this because Joanna didn't want to present this camp, because the brains of these individuals are all built from the same generative model, from the same genome, right? So we couldn't test this because we didn't have monkeys with different genomes or mice with different genomes. But so what we did, and this was Joanna's work, or one of many of Joanna's contributions, was to test this hypothesis by building classic vanilla recurrent neural networks that she trained to perform the monkey center outreaching task. And this is the usual methods that everyone has shown. So we could train the network to do the task, and you can see here the motor output. So it's beautiful, looks like a monkey. But the interesting thing here now is that Joe had the idea of what I'm going to do is try during training to have networks that produce latent dynamics that are more and more dissimilar to those generated by this standard vanilla RNN. So what Joe did was add a parameter to her cost function, alpha, that she could tweak, and as alpha became larger, then the latent dynamics of these networks should become more dissimilar to that of the standard network. And we can talk about that later, about the details. But the interesting thing is, if you look at these networks, you can see that as alpha increases, they kind of develop some tremor. But if you look at networks with the same value of alpha and you look at the correlation in the kinematics, they are all comparable. So all these networks within the same level of alpha are performing the task equally well, and the same is true for the errors. But the cool thing is that if I now plot the canonical correlations between networks for different levels, values of alpha, as I increase alpha, these canonical correlations become much and much lower, okay? So what you see is that you have the same behavior, but you can make the latent dynamics more or less similar. And Joe was able to do this even in a simple vanilla neural network with adding one parameter to a cost function. And actually, when she went back and looked at what happened, why these networks were different, is that the connectivity structure became different. So to us, this suggests that behavioral similarity is necessary for the preservation of latent dynamics, but not sufficient to fully explain it. But of course, we have some ideas to explore this in the future with animal data that we can discuss.
Sorry, was that in the alpha 0.5, the lowest curve there? Is this the average alignment between the dynamics of two different networks trained with alpha 0.5, or between the standard network and one alpha 0.5?
This is between two different networks trained at 0.5. At
that level, okay.
Yeah.
So there's more individual variability between networks in this.
Exactly.
Yeah.
Okay.
Yeah, this is, I think this was very cool, because what my intuition that I had never thought about, but I think Joe had already, is that as you increase this alpha, you are basically pushing the networks to explore in different directions of the gradient by adding these extra constraints, so their solutions become more variable, whereas the standard networks, they are always the same, which is also interesting for the, I guess, neural network aficionados in the audience. Yeah, so I guess we wrap up. So I started the talk a bit with our usual neural manifold spiel, but just to frame how we came to think about this question, or an answer to this question, I should say, and yeah, and a few other questions that we can also discuss if you want. I should also say that we'll have an updated manifold manifesto coming up soon with Matt and David Canaray. And focusing on this paper, led by Mostafa and Joanna. I've shown you that different individuals from the same species that are engaged in the same motor, or if you want, covert task, generate preserved cortical and striatal latent dynamics. I've also shown you that we've also shown you that these preserved latent dynamics are behaviorally relevant in the sense that they allow predicting behavior across animals virtually as well as you can within animal, or almost as well. And we've also shown you that our RNN simulation suggests that behavioral similarity is necessary but not sufficient to explain our results in principle. And since we have discussion time, because the talk is shorter than what Medad asked us to prepare, I figured I could add a few future directions that we had in the paper, like studying differences in neural activity underlying animals that perform the same task with different strategies, which may be easier to do in the cognitive domain than in the motor domain, because at the end, the motor output, if the motor output is very similar, it's hard to make the motor output different. Also, identifying differences in neural activity after disease onset, if you do different causal manipulations, or exploring similarities and differences across the species. And I guess with that, I would like to thank, of course, Joe and Mostafa for the excellent job, Matt, who has now joined, and Junchon and Josh and the for being on the paper, and everyone in the lab, and the people who've given us the money to do this. And of course, to all of you, first for inviting us, and second for the super lab email list that we all follow. Yeah, it's a great service to the community. Thanks, guys.
Questions?
I can ask one question. So thank you for the presentation. So I have a question that basically, so the first PCs basically has the highest correlation basically between animals. Like the question that I have is that, so you did the analysis of finding how overall the neural activity can explain the behavior, but do we see the same trend in terms of correlation between the ability of each of these PC individually can predict the behavior in terms of their basically so in other words, if one PC can predict the behavior really well, would we see that that specific PC also has a higher correlation in terms of animals, or we haven't basically mapped the behavior to each PC individually, or yeah.
Do you want to take this, Joe?
I think we haven't specifically looked at every single PC to see how much they explain of the variance, but we've also been really robust in looking at all kinds of different manifold dimensionalities to see if it's just the specific dimensions that we were looking at that was affecting all of the results. So I'm not sure if you can see this. Is it still being shared?
I think
Yeah.
Okay. Yeah. So here you can see we've looked at all kinds of manifold dimensionalities to see how it can predict. So we can see at the very beginning, with one PC, we get really poor decoding because I guess at least need 2 dimensions to explain the task. But then afterwards, you have better decoding. If you think it's only 2 dimensions that are needed, then you just need a 2-dimensional manifold, maybe, but then that doesn't explain a lot of the variance. Maybe it explains 50% and so on. So you do need a lot more dimensions, but after a while, it just plateaus.
Mm-hmm. So I guess my concern was, my question was mostly related to, so it might be that there is a small inter-animal correlation between some latent dimension. For example, I don't know, PC5 had a smaller correlation than PC1, but that PC5 might be able to predict the behavior better, in terms of some metric than, I don't know, second PC that had a higher correlation between animals. So I was wondering if you have any intuition on that.
I think there's a bit of a misunderstanding here that I kind of didn't explain well, because when we do CCA, I'll go back to the slides. When we saw here the correlation as function of the dimensions, here, well, you can see this doesn't mean that it's the same PC, because CCA is doing the rotation, right? So the axes that you find with CCA are not the same original axis.
But you could use that canonical vector.
Yeah.
And
Yeah.
project the neural activity on that vector and then look at the code. to answer
Yeah,
that.
which is not what we did here, but we've done it in another paper. So what we did it in this paper, in the Nature Comms paper. I think it made it to the paper. Well, the answer to this, what I can tell you is that, and this is also a paper from Matt Kaufman, when he was with Krishna Shenoy. The first PC that you find in these recordings, it tends to be like whether the monkey is moving or not, and is independent of on the details of the movement. And this is the first PC, the PC that you align best across monkeys. So the PC
Yeah,
that
that leads me to that leads me to my second. I mean, that's kind of the question that I wanted to ask. It's like when you decompose, you can. I mean, so if you decompose the variance in the neural variance first into what's common across movement directions and what's different between movement directions, and then do so basically in a DPCA you could separate the two, right? So you have a time dimension that could be more dimensions than one, because it overall that common dynamics could be. So is it the common dynamics that is more similar across animals, or is it also even like the directional specific dynamics that you get? Is that also similar across animals?
Yeah, that's a very good question. We haven't done precisely that in the multitask paper. We did that analysis, John. So most of the variance within task was unrelated to the target.
So within task is like relative to time and trial, right? I mean, that's the time in the trial is the main source.
The time in the trial is the main source, and then the target. So my intuition here is that we can probably the dimensions that we align best based on how well we can decode after a few PCs, but our inability to decode extremely well from the first two PCs is that the best we are aligning is two of these large movement independent PCs, and then the PCs that actually allow us to do the decoding. But that is a good question. Like we should try that out.
Because if it's just like the moving versus non-moving PC that aligns across animals, your paper would be a little bit trivial, right? Because then it would be just in both animals, motor cortex becomes more active when you start moving, right?
Yeah.
So it's a little bit,
but then you wouldn't be able to decode the continuous kinematic.
You wouldn't be
you would only be able
as an extreme point, that's the main point.
I know.
Yeah.
But yeah, this is what you see here. Like as Joe was saying, you probably need like 4 or 5 PCs to plateau. But it's interesting that it's more across monkeys after alignment, slightly more than within monkey. So this suggests that we are aligning across monkeys is a combination of these movement independent signals and movement related signals. Yeah. Matt, if you want to add anything, just hello.
Yeah, I mean, I generally agree with everything you said. I mean, the directional components have to align, or else you can't do the across decoding. But I also think it's interesting for this panel B here, where the within actually, if you squint, maybe like hits that knee and saturates faster.
Yeah.
Which I think of maybe as if it's not purely linear, it might actually take more linear dimensions to find the right full alignment between these two monkeys. Maybe there's like some curvature within the monkey, and if you do it within, you can do it with a couple dimensions pretty easily. But you kind of have to sample more of this space with these linear dimensions to get them across monkey.
Yeah.
I also have a question, and that is in regards to if it's useful to dissociate the geometry from the whole dynamics. So I was thinking like here you beautifully show that the geometry of that dynamical system is very similar across animal, but like you can have the same geometry but different dynamics essentially. The most extreme case would be like one fixed point that in one case is an attractor, but in the other case is a repellent here. So I was thinking that, first of all, is there a way to add the overall like timing and the dynamic of the system in the analysis? And if that exists, do you think this whole dynamic would also be similar across animal or not?
Yeah, so this is really related to one of our other supplementary figures. Let me see if I can find it. So essentially here we are like caring about the dynamics because we're using all of the time points and so on. But is it this one?
Yeah, okay.
We have moved these figures around so many times. I have to take a while to find them. But yes, so what we did here was essentially, what if we took away the dynamics completely? So what if instead of looking at like the time variant dynamics, what we did was like for each trial, we just found the mean of the activity in neural space. And here you can see what we do is now if we just align the topology, which is like purely the geometry and not the dynamics, then you lose a lot of the information. So here we can see when we look at the canonical correlations, when we're aligning the topology versus when we're aligning the dynamics. And here you can see for the same, you lose a lot of the information because it's below this line. Does that sort of answer your question?
I think he's also asking, like, even if you have the time in the trajectories, I mean, there's a geometry to the way that those trajectories carve out or go through the space. And CCA is largely aligning that geometry. So it's not in the paper, but many of you would know Olivier is now here in Montreal with Ming Guillaume. So he's actually used dynamical similarity analysis on these exact data. And DSA from recent paper from Ula Fiet's group is supposed to basically look for these type of fixed point line attractor type structure independently of what the actual geometric representation is. And we basically find that the across monkey alignment seems even more consistent, if anything, as if it's like really very similar underlying dynamical structure here.
Yeah.
But that's of course unpublished, but maybe if you come to NCM, you'll get to see it on a poster.
Thank you.
That method is, I think, a very interesting direction. We're also exploring it, something similar with dynamic time warping. So you could get into that. Like basically, you are going to see that you need to warp the trajectories very little across monkeys or across mice, animals.
So here you use the linear time alignment of the trials somehow to. How did you across monkeys? How did you align the neural trajectories in time?
In the paper, you mean, Jar? In this paper?
So for these analysis in this paper, you just did movement onset and then you stretched them somehow linearly.
Yes. So here, if I remember correctly, we were using.
I think it means in general, like we
Just
did
the main analysis of the paper.
movement onset.
Movement on.
Yeah, we aligned the movement onset.
And then a milliseconds after that. So
Yeah.
not adjusted for movement.
Yeah.
Just, okay.
Yeah.
Okay.
We didn't do any time warping, by the way.
Yeah.
Okay. No stretching, nothing. Just aligned to movement onset. Okay.
Yes.
Yeah.
And we don't do any trial averaging either, so it's all single trials kind of concatenated together
Sure.
for the linear alignment. So then, even within monkey, there's quite a lot of variability in like reaction times and trial lengths that sort of were kind of blurring together by doing this analysis in the concatenated trials.
Yeah, plus the variability in the instructed delay period, which was variable. And then the mice have their own variability of, only having done this for a couple of weeks and pulling on a joystick, not being on a lever, not being very mouse life-like.
So a question related to this is that came up is the mice are less well aligned to each other, but you could decompose your variance into the trial by trial variance within a mouse and then the mouse to mouse differences, right? So if you just take the average trajectory for each mouse, obviously the variability between mouse is also influenced by your measurement or the trial by trial variability that you have. You can decompose it, right? And when you decompose it, is it that the mice are actually, after accounting for their trial by trial variability, are they actually more different to each other than the monkeys? Or have you looked at this? And then, I guess the main difference has got to be the trial by trial variability that you're gonna have, right?
Yeah.
What about the true interspecies? You can estimate the true individual differences in the mice and the monkeys separately, right?
Yeah, I think that's definitely really interesting. We haven't done that explicitly. I think we just looked at total behavioral correlation across mice.
Yeah,
We-
that conflates both of the sources. Could be trial by trial variability, right?
Yeah.
Each mouse
Yeah,
does
yeah.
Fair.
thing, but they're just like random trial by trial, each of them. Or there are, you know, they have some variability, but like the mice generally have just more different strategies to do this task or different methods, basically, right? And I wonder if you've tried to get your head around it.
Yeah, that'd be very interesting to see if you have like more stereotype, like if you have mice with additional training, how that would differ in the analyses as well.
Yeah, so they do use different strategies. So if you look at, we haven't quantified this a lot, but we are doing some more stuff now. But if you look at the mice, I mean, this plot is a bit hard to see. But in this one, in mouse 40, like you can see that the ridges, the dot is where the ridges start, if I recall correctly. So you can see that they start in different positions for different trials. And what it seemed to be the case, looking at the trial by trial start position, is that, for example, this mouse always reached from here, and then when the joystick was somewhere else, he was kind of like adjusting little by little the reach trajectories and the initial positions. So it was
Palettes are two different palette locations, or what are they?
are different two different target locations, which is why the orange blob and the green blob are in different parts. So one of these mice seem to be like kind of the first reaches to the new location, he would be like kind of aiming at the same spot and then doing this instead of doing it directly. So it was doing like within block adaptation. So I think some of these mice also were changing the way they reach in the same condition within block even. Yeah, so this is something that I think it's more explored in Jun Cho's paper to some extent. I should have the reference for that. No. Yeah, more questions. I guess I can flag the paper while you guys ask more questions. Yeah, this is the paper that does a lot of description of this task for anyone interested. This one by archive. Yeah, but you have more questions about the paper that we're supposed to be talking about?
I was curious about the cross species differences between monkeys and mice, and so I might miss the point, but what if the dimension of the component would be increased for mice? Is that the decodability is increased as well, or kind of if you kind of maybe latent dynamics is increased, what happened?
Yeah, so I think so the number we use the same number of dimensions, right, for mice and monkeys, but basically the decodability plateaued before that.
Yeah.
So
Okay.
we have here somewhere. I guess we have it here, but not for the mouse. Or do we have it for? We do have it for the mouse. Oh, this is the alignment quality.
Okay, even if you increase the dimension, it's still kind of below the level, from the monkeys. So yeah, it's maybe related to the John's question before, but what the key point of the kind of difference between the mice and the monkeys is kind of could be ascribed to the structure of the kind of motor cortex of the mice and the monkeys, because its specification and the structure is a little bit different, because the monkeys may be more kind of segregated into the different part of the body. What do you think about it?
No, I mean, I think the main, I mean, there's two arguments, right? Like one may be that this task, if we believe in simultaneous cortical and subcortical control of movement, which I guess like there's evidence, and I do believe in that personally. Maybe mice rely more on the subcortical systems than monkeys. So maybe in this way, maybe in a sense, monkey motor cortex is more tightly linked to the kinematics of the movements than mouse motor cortex is.
Because my spinal cortical projection to spinal cord would be totally different, because it's more dorsally connected from in the mice, right?
Yeah, and also like there's a lot of like much more like innervation from brainstem, I think, even from basal ganglia output, like more significant, right? So that could be like one of the reasons. It would be interesting to see like what Joanna was saying, like we train a mouse until it's like a pro in the joystick, and then we can start to disambiguate also John's point about within subject variability. Yeah, to monkeys. So if we get a mouse to do the joystick task as well as monkeys do as well as the worst monkey does the reaching task.
Yeah, and personally, this might be a whole nother like PhD or postdoc, but I'm really interested in here. I think a lot of people, when they read this paper, initially they think that we actually aligned between the monkeys and the mice, but we don't do that. We actually just align within monkeys and within mice. But I think it would be really interesting, as you said, like obviously we have these structures that are evolutionarily preserved to some extent, even across species. It'd be interesting to see if we can notice differences between different species by saying, if we can align a mouse with a rat, would that have better alignment than like a mouse with a monkey? And so on, to differentiate like evolutionary components that preserved. I think that's quite interesting.
I don't know, the paper is kind of misleading, like, across animals, so actually
Across animals,
I thought
animals,
kind of,
not species.
oh, all right.
Yeah, we don't say across the species, we say across animals.
Okay, I have another question. So I was quite surprised that the extended data figure 6C, because in striatum has a similarly preserved, motor cortex and striatum similarly preserved. It's exactly almost the same. So do you think like is the other reason, like prefrontal reason, more like higher level reasons, or similarly preserved like this? And then I was quite surprising. I mean,
well, I think yeah, this at first this surprised us, like I guess like in 2021 or something, but actually it's good that I opened this, as a former monkey person, or still like former monkey chauvinist, I guess, until we started to work with Junchon and Joss on this data of the paper that I flagged, because they built this task, and then we started, what we expected to see in a striatum was not a lot of information about behavior, right? About kinematics and forces. But if you look at this paper and actually look at the ability to classify trial types of force kinematic interaction between motor cortex and striatum in the mouse, striatum is slightly better. If you look at panel E here, our ability to decode kinematics during reaching and pulling from motor cortex in blue and striatum is comparable, or if anything, striatum is slightly better. So striatum seems to have a lot of information about the details of the kinematics and the forces, at least in the mouse. So I don't know what this means. Like in this paper, we talk about joint specification, and that these representations may be these activity patterns may be the same for the same behavior, but they may serve different roles. These two areas may serve different roles during learning a new condition.
okay. What's the title of paper? So co-joint, I forgot that.
Conjoint specification of action by neocortex and striatum.
All right. Okay. Thanks.
You can also go on my Google Scholar justice.
Right.
But the nice difference that I want to flag in this paper, since you are talking about why striatum and motor cortex is different, is that if you look at this very final figure in the paper, you have here all the mice that are in two of these mice are not in the paper. If you focus on the panel on the left, what you have is the numbers of days that these mice have practiced on the x-axis, and then on the y-axis you have how well Junchon could decode the force that the monkey was, the mouse was exerting on the joystick. Sorry, and you can see that there's a striking correlation actually between how much practice they have and how poorly you decode force from motor cortex. So after 1 month of training, you decode force much worse than when mice see the force for the same time, for the first time. And this is not trivial, because if you do the same analysis for the reaching angle, you don't see this association. So we think that this is because maybe early on, cortex is doing all the doing, and over time, basal ganglia is taking like some of this load in this cortical subcortical, you know, I don't know, battle for control, or I don't know how to call it. Did this make sense?
Yeah, I would be very consistent with Olvetsky's, with Benzi's paper, right?
Yeah.
Exactly. It's very consistent with a lot of his results.
Yeah.
Okay, let's go back to the paper.
So we're at 2 p.m. if there's no other questions. Yeah.
Not so much of the question, but just in science fiction here. So how much data could you, I mean, okay, so this is a stupid idea, but if you had enough rich tasks across animals, across humans, monkeys, mice, you could do representational alignment between regions, right? So you could ask questions like, motor cortex in the mouse is this more like SMA or premotor cortex in the human or in the monkey, basically.
Yeah.
Do like a cross, you know, like compare structures not genetically but representationally, right?
Yeah.
If you think that's a good idea, I really hope you're one of my NSERC reviewers, because that's exactly what I want to do.
I love that this is on record on YouTube. Yeah, I think this is one of the things that we want to do, right, Jorn? So I guess like since also we are on YouTube, like we are excited to collaborate with people on these things, disease models, you know, cognition, different species. Like I know some of you guys are working with marmosets, for example. We could compare marmosets and macaques and mice, also like addressing like Joanna's point. So I think this is just like the first step in a very exciting direction where we can compare, for example, functionally after disease across the species and many other things, right?
Yeah.
Yeah. Thank you very much. That was a great discussion.
Okay, thanks for having us, guys.
Thank you.
Thank you, and see many of you in Croatia, I guess.
Yeah.